Unmuted. Excellent.
Unmute, yeah. Screen sharing now, or
Yeah, you can share your screen.
Okay. We were unsure if you would first like to have the introduction.
Excellent. Thanks. Okay, time to start. Welcome to this seminar in Ecology and Evolution of Montpellier. Just a few words before starting. So as usual, for those who are online, you are in a webinar mode, which means that your mics and cameras are automatically switched off. If you have any technical issue, please report to us in the chat and we will try to help you to solve the problem. If you have questions for our speaker at the end of the talk, you can use the Q&R or the Q&A tab to ask your question, and then we will allow you to unmute and switch on your camera such that you can ask your question directly. And if you are too shy, we can even ask the question for you if you ask us. Before starting, another word. So maybe you have seen we have launched a new call for proposing speakers for the seminars from October to next February. One specificity of this call is that we are switching funding. So the LABEX CEMEB, who was funding the seminars, is stopping at the end of the year, and this will be taken over by the PTL3, Programme Thématique Long, from Pôle AEB. And this means that during these 4 months, roughly, we won't have funding. So we ask you to propose speakers who are already invited through projects, etc., or local speakers. So this is the specificity of the call for next month. And now I leave you the floor to introduce our speaker. Thanks a lot.
Yeah, hi everyone. I'm really glad to introduce today Nico, who was my PhD supervisor, collaborator, and friend. So Nico did his PhD some years ago, not so long, in Darmstadt, in Germany, working mostly on plant diversity and ecosystem functioning. And yeah, he moved a bit to Minnesota and then came back to Germany with professor now at Leipzig University and iDiv, the German Center for Integrative Biodiversity Research. So yeah, a lot of projects are to summarize in a few words, a lot of prizes, but this is one is the most proud of is winning the soccer cup in Leipzig.
Did not win.
So yeah, I just leave you the floor and we'll introduce it as a highlight.
Merci beaucoup, Rémi. Je m'appelle Nico, j'habite en Leipzig, en Allemagne. Je ne regrette rien, and this is what I can say. Sorry. Yeah, thanks a lot for inviting me, Rémi, Stefan. Thanks for coming, for the interest. Today I would like to talk about the dark side of biodiversity, a really broad overview of the work that we are doing. You will see that the videos that are implemented in the presentation are a little bit slow. Maybe this is a good fit because yesterday we had some really good wine tasting. So maybe my mental state and the videos fit together and we can keep up. So when I'm presenting results, it's not only my work, it's the efforts of many people, also several sitting here, and yeah, lots of local and international collaborators. Many of them are happy. Yeah, we are in Leipzig, where I have a professorship for experimental interaction ecology. We are looking at biodiversity from very different angles. So we look at the biological diversity outside, trying to describe it, trying to understand how is it changing, why is it changing, what are the ecosystem consequences. Also for us as humans, what are our inputs and impacts. And as you can see, this is a powerful center, but some parts are missing. We are now moving from funding by our main funding agency to institutional funding, and some of the pieces are not there yet. We're looking for alternatives, but still it's functional. And yeah, we are only one of many groups at iDiv. As I said, experimental interaction ecology, which means that we are performing experiments to understand interactions between species, and we are broadly interested in ecology. We made up this beautiful logo here, which stands for our three main research pillars that I would now also like to revisit in this presentation. First is, you can imagine that this is representing a globe. We are interested in the global distribution of biodiversity. Why do we find more species here and not there? And we mostly do this below ground, look at microbial diversity, at animal diversity. And then you can try to understand the potential drivers of this biodiversity. So how important is the role of different land uses, climate, soil conditions, and so on. But what you will get from such global studies, from observations mostly, is correlations. And you cannot really be sure about causality. And this is where the ecological experiments come in, where we can manipulate things, where we can study the drivers of biodiversity. We can heat up the system, we can remove precipitation, change land use, type and intensity, and all things like that. And you can also imagine that these portraits here are representative of experimental plots where we apply different treatments. Higher temperature, more precipitation, lower precipitation, and so on. And then we can look at the ecosystem responses, at the biodiversity, and also different ecosystem functions, how much carbon is stored in the system, how much biomass is produced, how are nutrients recycled. But again, yeah, you get a biodiversity response potentially and an ecosystem function response, but you don't know what is actually the role of biodiversity in this process of ecosystem functioning, because in such a context, you can also only look at correlations. And this is why we also manipulate biodiversity and do biodiversity ecosystem functioning relationships. We can, for example, manipulate the number of plants, of trees, for example, that co-occur and interact with each other. And you can then study how do they perform, what are the different processes that are going on. And coming back to this logo, you could imagine that these are now different species in a food web where different organisms feed on each other, interact. There are different kinds of interactions happening. And we look at these biodiversity effects. Yeah, you may ask, why the dark side of biodiversity? Why are we looking into the soil? There are so many colorful and beautiful things out there. Why do they look into the soil? And this is one important figure in my life. Master Shock. Stefan knows him. Yeah, he was quite influential. He told me during my studies that the force of the dark side of biodiversity, consider you must. Sorry for the grammar, but I think it's more or less how he talks. So when I studied ecology and biology in general, I was really fascinated by his lectures and also excursions, and he then also introduced an exciting topic to me, and this is an invasion event in North America. This is nice photo from the Canadian Rocky Mountains, and in this region there is no native earthworm fauna, because after last glaciation, native earthworms did not recolonize these regions. They're just too slow. They can cover maybe 3 to 10 meters per year, and then recolonizing thousands of kilometers of the whole continent was not feasible for these small creatures. So huge ecosystems without any earthworms. And you can have beautiful forests with a lot of understory vegetation, high biodiversity, before earthworms invade, because these systems adapted to the absence of earthworms. And if earthworms come in, and this is mostly European earthworms that we introduced there on purpose and also accidentally, then they turn the ecosystem upside down. They change conditions so much. And this was for me like an eye-opener where I understood, okay, this below ground biodiversity is so important for the general functioning of ecosystems. This needs to be explored in more detail. Common question, which comes here. So maybe I get for 5 additional minutes at the end because I'm already responding to one of your questions. Earthworms are good for most of our plants where you have a poor evolutionary history between plants, organisms like microbes and animals and earthworms. So be happy about all of the earthworms in your garden, but in conditions where they're not native, they can just change the conditions in a way that organisms are not used to and also induce some biodiversity loss. And you can also imagine that the whole nutrient cycles are strongly altered. So now let's move into the soil. I will now bring you below the ground where my most favorite organisms are. Sorry for the video. You can see some earthworms and here microarthropods, different kinds of protists and microorganisms driving essential ecosystem processes. Decomposition is one that is also in the focus here locally. Stefan and his team have worked a lot on that, world-leading experts to understand this process. And for me, it's always interesting to look at interactions between these different kinds of organisms and important functions. This biodiversity can also be beautiful and interesting. This is a tardigrade, one of the toughest animals, or the toughest animal on Earth. You can even bring it to outer space and it will survive at minus 260 or whatever for some time in vacuum. You just crumble together, and then when you bring it into more appropriate conditions, it will inflate again and continue living. This is nematodes. 80% of all metazoans on Earth are nematodes. So if we talk about biodiversity, I think we should talk about nematodes. Many of them occur in the soil, or most of them. They cover all kinds of different trophic groups. You can have microbial feeders, plant feeders, predators, so a whole micro food web just composed of these tiny but important creatures. This is an orobatic mite that also plays an important role in decomposition processes. And some cute animals like here, some species of Collembola. This is also Collembola or springtail. So that can be nice and interesting. And you can also have terrible predators. This is a predatory mite feeding on a beautiful Smintovide Collembola species. And the most favorite one, the earthworm. There have been a lot of studies now and a lot of recent interest into the drivers of biodiversity. And you might have heard of the IPBES, so a consortium of scientists and also different stakeholders that summarize information on the state of biodiversity for decision makers. And what they could summarize is, for example, here, some indirect drivers, but also direct drivers of biodiversity in different realms, from terrestrial to freshwater to marine. And you can see that land use change, direct exploitation, climate change, pollution, and invasion events are really important drivers of biodiversity. You can also potentially read some of incredible numbers. Some of them are very scary. But what these studies looked at and what these people could summarize is mostly based on plants, mammals, fish, also some insects. But basically this assessment stops at the soil surface. And they miss almost 60% of the total biodiversity on Earth, which is in the soil. So this is quite a substantial fraction of global biodiversity. Let's try to understand it. And a couple of years ago, we also contributed to a global assessment of the state of soil biodiversity. If you're interested in that, you can just download it, where we try to summarize the current state and also potential future research needs. So I said, yeah, we know something about changes in biodiversity, mostly based on these creatures here, mammals, fish, plants. You can see here the pie charts representing the expected number of species within these groups of organisms. And you can also see the expected fraction of described species. So the described proportion is displayed in this green color, and the undescribed part is in black color. And you can see that mammals are really few. There are really few species. They are very well described. Even for fish, there is some work to do, and for plants, still biodiversity to identify, to describe. If you then move to insects, yeah, it's roughly about a species, maybe for some taxa less. And now let's move below ground. For example, earthworms. There are more earthworm species than mammal species. At the moment, we have roughly 7,000 earthworm species described worldwide, and we expect that more than 20,000 additional species are to be found. This is, for me personally, unbelievable. And it gets worse when you look at Collembola, more species, less described, and then nematodes with only 0.2 to 2% of the species being described so far. So a lot to do. If you're unsure what to do in the future, you can potentially study nematodes or other groups of organisms. Where should you do that? There are some blind spots globally where we know very little. You can see, if you look at different biodiversity groups here, ranging from microbes to earthworms, or if you look at different ecosystem functions, and this is here a representation of below ground biomass assessments, that we have typical hotspots of information. We know relatively many things about Europe and North America, for example, but there are also these cold spots where we know incredibly little, often in South America and Africa, large parts of Asia, where we have not been able to compile respective information. Then the question is, if we know so little about below ground diversity, can we just use information on above ground diversity, which is much better covered, to predict what is happening below ground? And we call this the Beauty and the Beast project. So can you use information on the beauties here above ground to predict what the beasts in the soil are doing? This is maybe a less appealing creature of a mite. So we try to do this, and using a very simple approach and taking published data from mammals, birds, amphibians, and plants, and put them all together. So a really blunt and rough approach. For each group, we determined a biodiversity index, and then we all put them all together. So they're all created equal in this approach. And just to see how are they distributed if you have one above ground biodiversity index. And then we compared this with some below ground information that we had at that time. Yeah, in the meantime, many things have happened. I think we're getting more, much better now in terms of data availability. At that time, we had mostly information on the global distribution of bacteria, fungi, and some macrofauna taxa. And then you can look at matches and mismatches of these different biodiversity patterns. So we have here now a colorful map where we plot soil biodiversity against above ground biodiversity, and you have a scenario where both is low. This is represented in white, easily to understand here. For example, in the Sahara, there is low below ground and low above ground biodiversity. Then we have regions where both is high, many regions in the tropics here. But then you have also a lot of yellow and blue areas. yellow representing high above ground and low below ground biodiversity, and blue representing high soil biodiversity and low above ground biodiversity. And you can see a lot of these areas. So we cannot really predict. There are lots of matches, but also lots of mismatches in the distribution. And it's really important to have good information on below ground diversity. We then contributed and led a global synthesis of earthworm data. This is more than 10,000 data points that we could assemble for earthworms and then look at the very local diversity of earthworms. We could create global maps of earthworm distribution. What you can nicely see is that earthworms like the UK because there are a lot of sheep and there are many species of earthworms like sheep poop and pastures. But this is really rough information. At the same time, it allows us to identify some of the main drivers, as mentioned at the beginning, that are important for different indices of earthworm communities and diversity, like their biomass, their abundance, and also species richness. And here in a very simple analysis, we just compared the significance of different drivers. You can see that habitat cover is very important for biomass and abundance, maybe a bit less so for richness. Precipitation is very important for earthworms in general, easy to understand, but also meaning that if we experience any changes in precipitation, this will have significant consequences for below ground biodiversity. We are now in the process of starting the second phase of a global sampling campaign in Soilbon, the Soil Biodiversity and Ecosystem Functioning Observation Network, where we measure many different variables across the globe from different locations with a huge network of collaboration partners here. And the main aim is to do measurements in a very standardized way. The data that I showed you before was all coming from different sources, different methods being used. Here we do all measurements in one lab, very standardized way. And we would also like to do it repeatedly to monitor biodiversity changes, potential changes. And then if you have a temporal trend, you can also relate this to the drivers that might have changed. Why is this important? Because we would like to also protect this functionally important biodiversity. We'll come to this point a bit later, how important this is. But we need to get active already without knowing how this biodiversity develops right now. The question is where and how to protect soils. The how is presented later. The where, you can come up with different indicators. And this is the most colorful figure I've probably shown in my life. Maybe you cannot derive a lot of conclusions from that one. It looks like we're taking drugs, but what it should represent is just very different indices that you can derive from below ground information. You can use, for example, sequencing data on soil microbes and animals to assess their alpha diversity, which means the very local diversity below your feet. You can look at community dissimilarity. So is it the same species here under my feet or there? So for example, in the tropics, you have really high dissimilarity among the communities in space because there are a lot of different conditions. While in temperate regions, you might have more homogeneous conditions and less community dissimilarity. And then you can also look at different ecosystem functions and services that are provided to us. I already mentioned carbon sequestration, but also different nutrient cycles and so on. And then you can try to understand how is this distributed globally, where is one facet more represented than others. What do I want to protect, in fact? And this is just one exercise. This all depends also on our preferences and decisions. And what is it useful for is you can then also define some priority areas where maybe many of these variables come together. And you can go to the more brown regions to then select conservation areas if you to select some new areas. Rarely done on the basis of soil biodiversity, or never done. Maybe this is the first idea how it could work. So I don't think that this is the final solution to this problem. It's just an attempt to facilitate decision-making, to consider the multidimensionality of soil biodiversity. Why is this? Yeah, there is an intrinsic value of biodiversity, I think, but in addition to that, we have benefits, and we can study the benefits of soil biodiversity. So let's look at the main drivers first. And again, I would like to come back to this figure that was produced by iDiv. What you can see here is that these drivers are only assessed here in isolation, but they all co-occur. Yeah? They are not happening in isolation. And maybe there are also interactions happening that in fact is more pronounced when you have climate change happening in an area with intensive land use and where there's also pollution happening. Maybe you have non-additive effects, so more than you would expect. And actually, I was involved in a survey of more than 3,300 biodiversity experts, and yeah, the huge majority of them is totally convinced that it's about synergistic effects of different drivers, so that we have to study them together to better understand how biodiversity is changing. In addition to this complexity, soil is quite difficult to study. We mostly take snapshots. We go out, take a soil sample, some colleagues sequence it. I can count the earthworms or measure some ecosystem functions, but this is then one measurement in time, and maybe you go out during the wrong time, or this is a picture showing you that snapshots are not always representative of conditions. This is, for example, a tour that we did with the lab group many years ago. We wanted to take a group picture, but this postman was in a rush. So this is my foot here. I'm still part of my group, but in the snapshot you don't see me. And maybe when you go out and do a sampling, you will also miss important. Maybe I should rephrase this. Anyway, so let's do multiple measurements. And this is not so easy because it's very laborious. If you would like to do biodiversity assessments, you have to spend a lot of time with a microscope or in the lab sequencing. And this is why we are very open to simple methods to test hypotheses. And this is what we did in this experiment here, which is a climate change experiment in Minnesota called Be Forewarned. In this experiment, we have different temperature treatments. You can maybe see here some infrared heaters. And then there are also these tarps where you can remove precipitation. And in this experiment, we have manipulations of both precipitation, basically a summer drought, and temperature. And then we take a very simple method. It's called the bed laminar method. And you can have some stripes with me if you're interested in that. So tiny PVC stripes with holes, and we can fill the holes with a substrate that is delicious to soil animals. If you put it into the soil, they will just start feeding on it, and depending on their activity, there will be more or less feeding happening, and then you will have a different number of holes appearing in this bed laminar stripe. So you can see it's put into the soil, and then you're feeding here mostly of microarthropods, and with that you can do a lot of measurements because it's a very simple method. You can use this in citizen science projects, for example, because everybody can count empty holes. So by counting empty holes, you can produce a lot of data. This is now data across 2 years of experiment, and we assessed detritivore feeding activity, meaning this feeding activity on the substrate. We put in cellulose, and animals fed on the cellulose. Here the activity is plotted against time, and in the different treatments we have ambient temperature, then plus 1.7 degrees warming, plus 3.4, and then ambient and reduced precipitation. If you look at that, you see not so much, a lot of points, a lot of variation. You see that in general, activity patterns show some temporal dynamics, so maybe it's good to do multiple measurements. And if you summarize all of this information, you can see quite interesting results, because if you, for example, look at the detritivore activity just affected by warming under ambient precipitation, then you don't see any significant warming effect. So all of these bars here are similarly high, and there's no statistically significant difference. But if you are now reducing also precipitation, a scenario which occurs quite frequently, so higher temperatures and precipitation, suddenly you see significant effects that there's the lowest activity at high temperatures with reduced precipitation. And this is a log scale, so the effects are quite substantial. So I think you now understand that it's important to look at such interaction effects between drivers. And we did this also in a global change experiment in or close to Leipzig, where we have different land use types. And the question is, can we somehow adapt? Can we mitigate the effects of climate change, for example, with more sustainable land use? And this is the main idea of this experimental setup, a huge experiment with 5 land use types. We have 3 grassland treatments. 2 of them are extensive, and 1 is very intensively managed. One is grown, and the other one is grazed. And then you have an organic farming and also conventional farming. And this you can see here again in this video, 5 land use types. And yeah, on top there is a climate change treatment. We have ambient climate and a climate that represents a scenario for 2070 for the region. It's highly replicated in space, and it's also, then you can see a tractor here. So it's really large scale, beautiful facility. And these roof structures can be used to, for example, increase the temperature. If you close them by night, you can reduce the loss of energy. And you can also induce changes in precipitation patterns, and this is the main aim of this experiment. There will be more precipitation in spring and in fall in the future, but much less precipitation in summer happening. So roofs can be used to remove the rain. As you can imagine, we collect this water in a lake, and then you can make the sheep wet in spring and in fall. So this is the experiment. And again, we looked across many years. I think we should now work on the next time series. There's now much more data, but what you can see here is that there are benefits of sustainable land use, but you have to be patient. Let's look at the initial years. The experiment was set up in 2013. So measuring after 2 years, you don't see any significant differences between these different treatments. The colors representing here farming and grassland systems, not all 5, only 4. Grass low and high intensity, and farming high and low intensity. And here, not so much, maybe even the higher soil microbial biomass found here in the highly intensified grassland use and also in the high farming system as compared to the extensive used system. But then over time, you see a switch, and you see also that they spread more, that the differences become larger, that the grassland is performing much better in terms of soil microbial biomass. And after some time, there's also this switch here happening between these grassland land use types, that the low intensity land use in grassland shows some benefits for soil microbial biomass. So yes, there are some benefits, but you have to be patient. And a similar analysis will come out soon, where we look at ecosystem multifunctionality. So this is now looking at a lot of different ecosystem functions in parallel. How much crop yield is produced, how much carbon is stored, how are nutrients recycled, how much nitrogen is lost also from the system, for example. And then you create one index of multifunctionality and can look at the effects of the different land use types and the climate. And here, if you look at the short term, you see the lowest values for future climate, yes, but mostly here for low intensity grasslands and pastures, which I would have thought would be really beneficial for ecosystem multifunctionality. So quite surprising short-term effects. But if you then look at long-term effects, you can see that this also changes completely, that the highest performance is found in low intensity grasslands and pastures, and that also these climate change effects are a bit more buffered. You see stronger deviations in the other treatments as compared to the pasture, for example. So it's very beneficial to have time and to follow such studies over time. How does this relate to soil biodiversity? It might be a main driver actually of ecosystem multifunctionality. We have now evidence from multiple experiments. And here we could see, for example, that the variability of agricultural yield, which is very important for farmers, decreases with soil biodiversity. So you have more stability in crop yield with higher soil biodiversity. This is a correlation. We need more experiments. So I will speed up now a little bit and summarize things in a bit more concise way, showing also some global evidence for the benefits of sustainable land use, because this is a local experiment. You can argue, okay, this is important for Leipzig, but Montpellier is so different, might not be beneficial. This is why we do a lot of global synthesis. Here we summarized information on soil biodiversity responses to different drivers in experiments, and we could see that if the mean and deviates from the zero line in a negative way, you have a detrimental effect of climate change on soil biodiversity. And if the error bars don't touch the zero line, it's also a statistically significant effect. And what we concluded from this global study with many hundreds of data points is that climate change overall has a detrimental effect on soil biodiversity, land use intensification, pollution, that is nicely shown, while the other drivers here are not that clear. Maybe even for nutrient enrichment, you see some more beneficial effects. And if you now zoom in, you can also identify the main drivers. For example, within climate change, it's mainly drought causing the detrimental effect. So drought is important for soil biodiversity. For land use intensification, it's inorganic treatments, it's tillage, which is most important. For pollution, it's everything. Pesticides, metals, it's all important in decreasing soil biodiversity. And for nutrient enrichment, you can also increase soil biodiversity by adding organic fertilizers, for example. This is what we could derive from the study. So I will skip this one. What can we do? We can manage our ecosystems in a sustainable way, I think. We saw that the sheep grazing, which I also understood yesterday during a short excursion, used to be quite important here in the region. Can be beneficial. Deadwood management is very important in forests, and we are working on different ways of trying to provide also recommendations for that. So now let's move to the last chapter, and this is biodiversity ecosystem function relationships. I always already touched a little bit on it, but now let's get at experimental proof. And this is a first example where we try to summarize global information on the importance of biodiversity for ecosystem functioning and stability. At iDiv, we have a synthesis center which allows working groups to come together and assemble nice data sets. Here we used a global data set of 46 different grassland biodiversity experiments. In all of these experiments, you have grasslands where different plant communities were assembled, monocultures, 2-species mixtures, 4-species mixtures, and so on. And then you can look at the performance of these mixtures. And what we saw is that overall productivity, this is now just the yield of these grassland communities, increases significantly with the number of plant species. And this is true if you look at wet years and dry years, and you see a lot of lines in the background. Those are the different experiments that are represented here. This was already interesting, but I think what was the most important outcome of this study was that there's also higher stability of productivity here assessed as the resistance with higher plant species switches. So it's more likely to get stable productivity over time. So skip this, although this is Remy's topic. Maybe I cannot skip it. How long do I have? 10 minutes. Okay, then I will not skip it. So this is one part of stability. Just looking at the biomass production of systems. But we have also nice experiments where we can look at the stability of other ecosystem properties, for example, of soil temperature. We have an experiment in grasslands where we have 80 grassland communities, where we have sensors in the soil in 2 soil depths, sometimes even in 3 soil depths, and we measured soil temperature across 18 years in this experiment every minute. So a really huge data set, and you can imagine that you can look at different stability facets, daily stability, monthly, yearly stability, and so on. How stable is soil temperature? Why are we interested in that? Because soil temperature is so important for many microbial processes that are going on. And what we found was that soil temperature stability is determined by the number of sown plant species in this experiment. And we also identified some of the main drivers. It's basically the coverage of the vegetation that with more plant species, we already saw in the previous synthesis that there's higher productivity, more dense vegetation, and this is buffering the system. It's like putting on a couple of pullovers. It will buffer temperature fluctuations. And in the soil, there's also significant changes happening, that there's an accumulation of soil organic carbon in the soil. And this soil organic carbon changes soil properties, the physical properties of the soil, and also contributes to soil temperature stability. So this is evidence from grassland, and this is similar results were found also for forest, where Remy was involved. Again, looking at biodiversity effects, here it's the number of tree species. This time, we did not look at the below ground temperature, but temperature in the understory. So, and a temperature that we would also experience when we go into the forest. And again, trying to look at the main drivers, you can see that the coverage, LAI, of the vegetation is increased with higher tree species richness, and this then buffers the monthly temperature here. You have additional effects that are mediated by the structure. The more complex the vegetation structure is, the more buffering you can see. So this is quite consistent findings across these very dissimilar data sets. And Remy knew it before, many years before this was done. He already said diverse forests are cool, so there is a cooling potential, for example, in summer. And this is also why you probably enjoy going to adjacent forests when it's really hot here. Why is this? So let's explain biodiversity effects. And I used to be a soccer player. Sorry, this is a German team now. Maybe I should have put Paris Saint-Germain and Kylian Mbappé, but this is an old team of Bayern Munich to explain complementarity. So different species in nature complement each other. They have to be somehow different, otherwise they would not coexist for a long time. And you can nicely explain this with a soccer team, where you have different positions and you need to have different traits, different skills to be good at these positions. This was a quite successful team. with a diversity of skills. And if you would compare this team just with the best monoculture, with the Robert team, for example, I would imagine that the left team with the different players would win. And this is what we typically find also in ecology, that it's really the difference in these traits that is much more important than this one trait that Robert or a certain grass species has. Yeah, we then looked also at the question, how important is biodiversity? And again, time matters. So I will briefly describe this here. In two very long-term grassland biodiversity experiments, we looked again at an indication of plant biomass production. And now you can see here in different colors, different experimental years. Early experiments or years in red and later years in blue. And you can see that in early years, it's a significant positive relationship, but it's saturating. And if you're losing species at the high end of the gradient, you would not really think this is important for ecosystem functioning. So you might come up with very different conclusions in comparison to a treatment here that is long-term, where every loss in species would result in significant change in ecosystem functioning because the relationships get more and more significant. Why is this? We are exploring this right now in different experiments. We think that it's very important that these plants don't occur in isolation. They have a diversity of interaction partners, and this is also where our most research interests lie, not only below ground food webs, but also now tackling above ground food webs and also the energy fluxes between these different players. So plants are embedded in this complex network of interactions, and over time, there are just changes happening in these experiments that cause high diversity communities to function better. So it's very important to have old biodiverse grasslands because you have a higher proportion of mutualist beneficial interaction partners, whereas at low diversity, antagonists accumulate over time. And this is also why farmers, they have crop rotation. This is biodiversity in time, basically, because they would like to dilute the accumulation of pathogens. And in these experiments, we see biodiversity in space. We are studying such relationships now in the Jena experiment. We have a huge research group working on this, looking at not only ecosystem functioning, but focusing on also more on stability. And we put a specific focus on community, assembly, and feedback processes. So more to come from this world. And then we also set up a tree diversity experiment that specifically looks at interactions between plants and microorganisms that are associated with roots. Mycorrhizal fungi play a really important role in plant nutrition. They form a close relationship with almost all plants on Earth, and the fungal partner receives carbon substrates, assimilates from the plant, and the fungal partner provides nutrients and water to the tree. And we think that this interaction is very important to understand biodiversity, ecosystem function relationships. So you can see in the background, this is the Global Change Experimental Facility that I mentioned before, and next to it, we now planted 80 forest patches to study such interactions. So to provide a final outlook, biodiversity is very important to consider for us as a society. You have seen probably many studies or opinion papers highlighting that there are important interactions between climate change, biodiversity change, and how I would like to focus here mostly on soil biodiversity and society. There are impacts, there are feedback effects of society. And I think we have to put this important fraction of biodiversity more in the focus of this research, because this biodiversity actually regulates how much carbon is in the soil and feeding back to the atmosphere, how much greenhouse gases are emitted, and so on. So very important processes. And I think the next important frontier is also to integrate across systems. We have see now mostly data and experiments either in cropland or in grassland or in forest, but these systems are connected, and I think this connection, this diversity in the landscape is really important to consider in the future. So coming back to these pillars, we now stay mostly within, but I'm totally excited when actually these different pillars meet each other and inform each other. And a very final example on this is where we combined observational data from Europe. You may have known of the Lucas survey, a lot of sampling locations across Europe doing monitoring of soil conditions and now also soil biodiversity and ecosystem functioning every 3 years. And we studied grasslands and croplands in this framework, and we compared this with our global change experimental facility, where we have also grasslands and croplands and different climate change scenarios. And then you can do comparisons to get at causal relationships. This is much too complicated because also it's the final figure, but the main conclusion from it is that we see similar patterns across the European survey and in this local experiment, that climate change, when it's reducing precipitation and mostly the precipitation change, is causing lower soil multifunctionality, so it's detrimental to soil conditions, and also an intensive land use is detrimental for soil biodiversity. And this is, I think, a nice comparison of these different approaches. So finally, I would like to thank a lot of funding agencies for supporting this. Yeah, there are many collaborators, so it's not only us. Many people are looking into the soil and data providers. I'd like to thank you for your interest and attention, and then may the soil be with you.
There's a really cool example from France. One of the first countries, I think, that published such an atlas of the field diversity and how it depends on different land use types. And I think this was also created for farmers to provide some feedback where they could also then interact and get information on their field sites and maybe adapt their management. What to do themselves, I mean, when I interact with farmers, they often know quite well how their soils are doing, how their earthworms, for example. That's easily done. And I think there are now many also citizen science methods out there that help assessing at least the functioning of the soils. I'm not sure if they have to measure biodiversity directly. They can do it with some selected taxa, like earthworms are pretty easy, and many are already doing that. But in addition to that, they are also interested in how well is it functioning, and then they could also look at some decomposition processes. So I think that soil carbon measurements are easily done themselves, but this would be another important indicator. But you know about these stripes and feedbacks, wood sticks that give you an indication of overall.
Thank you for the talk. I really enjoy it. You often speak about soil biodiversity, but my question is about, do you observe the same pattern for microfauna and microbial diversity, or opposite trend, or?
Yeah, it's quite the similar distribution patterns. We are working at the moment, again, our synthesis center at the global synthesis of different macrofauna taxa, and even within this group of organisms where you might expect similar responses, you find different distribution patterns and also drivers. And for the microbes, it's another different world, I would say, because you have different clusters of microbial diversity that were described also more in drier regions and in wetter regions and with high and low pH. So I think there are significant differences, and I put many things together often, was broad in my descriptions, but you're completely right that there are similar distribution patterns.
Okay. And do you observe sometimes like higher diversity of microorganisms with, for example, land use intensification, or?
Sometimes yes.
Yeah, sometimes yes. Yeah.
Bacterial diversity.
Bacterial
sometimes even higher when you have some problems that we don't really understand.
Yeah, we don't understand why. Yeah.
Okay. So you cannot help.
No.
It's quite surprising. But this is one group out of many. If you then look at fungi, they mostly not be affected by intensive land use. I'm not sure if it's also due to some competition for resources, carbon. There's less of the others. Bacteria groups are better at that. This is an open question.
Yeah, one more.
Just keep it.
Okay. Now, it's just about time. It made me think, because most of our experiments are for 1 or 2 years, but what you show is like time is very important. So I don't know if we can. Yeah, I mean, it's a very important point. It's more like an important point than a question, but maybe we should, that's for an experiment. It changed all the figure you show, changed everything. So
changes everything. It's true, and it's a challenge. And I think a great example, Alex who did his PhD in the same experiment as I did, the Jena experiment, when he measured at the very beginning of the Jena experiment, the plots were just established, what is the role of plant diversity for below ground organisms and functions, he concluded that there are no significant effects. And when I measured several years later, I found these effects, significant effects. And I think both information is really important because you have a lot of land use change, and it's important to understand when are and also changes happening, how can you facilitate changes maybe, but it requires platforms that are in place for a longer time. I would never discredit any short-term measurements. I think it's still very valuable, but it's important then to keep measuring, somehow trying to maintain, if it makes sense, maybe in a crop system where there's high turnover, maybe less sense, but forests, grasslands.
I have a question concerning politics. Well, you showed us the complexity of the system and the interest to to focus on this complexity. So how can we transfer this information to politicians now, because we are in the soil directive now, and you know the results of this for the moment. So they retain only very few parameters. So what can we do at this level as a scientist?
Yeah, I'm not sure if I'm the best qualified person to comment on that. You might be much more qualified and maybe thought about this much longer, but I think it's a challenge, yeah. At the same time, an important topic to tackle. So what we try to do is to try to summarize information as good as we can in reports that are concise and that transport important messages. I think the preparation of scientific information for a certain community is quite important, because they will never read any paper that I showed here. But reports like iDiv, IPCC do have an influence. We now have a project also in Germany. It's like the German iDiv, an assessment of species diversity across Germany, and this was even funded by the ministry. So there is some interest, and they also provided feedback on how to communicate. I just hope that, both sides show interest and also invest time. This is also for us a huge time commitment to then translate our findings into more accessible conclusions. This is the first critical step. And then I think it also depends on voters. So also do some public outreach to inform society on the benefits of action, for example. It might also influence how they vote, and politicians are more or less interested. But
we have more questions for everyone.
I had a very nice question, but the stuff that you showed about the find all around and below. Between these, what happens above or what happens below. Yeah.
I think we are looking at very different types of organisms with whole different life history traits. For example, if you go into polar regions, you will probably find less above ground biodiversity, but you have thick layers of organic materials that are processed by a diversity of decomposers. So it's different scales where these organisms also act on, and just then based on these life history traits, habitat requirements that drivers have. So we could touch on each of them individually, what is most important, but I think it's just also a matter of scale. Can we compare a panda bear and its biodiversity distribution to bacteria? I think this is an important topic that we still need to cover. I showed you mostly information on alpha diversity. If we look at beta diversity, I would assume that maybe the global distribution patterns would be a bit more similar. That most biodiversity in the topics.
Okay, thanks a lot. Unless we have another question, I think we can thank Nico for the presentation, and thank you a lot for your attendance. And we'll see each other next week, right?